An octopus arm lies against a rock on the seafloor, motionless in the way that water is motionless, holding still, but never entirely still, the faintest drift of current, drawing the tip of it sideways by a few millimeters and then back again. The rock beneath it is dark and cold. The arm rests along the stone the way a sleeping hand rests against a pillow, loose at the joints, the suckers pressed flat against the surface in a line. Nothing appears to be happening, and yet, along every centimeter of that arm, something is. The suckers touching the rock are reading it. Each one carries chemoreceptors that are sampling the mineral character of the stone, the faint dissolved traces of the organisms that have lived on it, the microscopic texture of its surface, the temperature that has been leaching up through the seafloor from deeper layers for longer than the arm has been in contact with it. The arm is not waiting for instructions. It is already processing what it finds. Signals are moving along the dense cord of nerve tissue that runs the full length of the arm like a thread of compressed thought, and the arm is already adjusting, a slight increase in sucker pressure here, a minor relaxation there, not because the brain overhead has decided anything, but because the arm itself has. This is where the story of the octopus begins, not in the mind above the eyes, not in the round, soft mass of the brain tucked inside the mantle. It begins in the arm pressed against the cold rock, in the quiet and continuous act of an intelligence distributed so widely across a single body that drawing a line between the part that thinks and the part that acts is something close to impossible. There is an animal with roughly 500 million neurons. That count alone is not what makes it worth attending to. Many animals have large nervous systems, and neuron counts are a rough measure at best. What makes this animal different is where those neurons live. Of that 500 million, somewhere between 300 million and 350 million of them do not reside in the brain at all. They run through the arms, 8 arms, each one carrying its own dense cord of nerve tissue, each cord capable of generating responses, processing sensory information, and coordinating movement without any signal reaching the central brain first. The brain, in the conventional sense, is almost a minority partner in this arrangement, present, coordinating, important, but sharing authority in a way that has no parallel among the vertebrates and very few parallels anywhere in the animal kingdom. To understand what this means, it helps to hold the comparison for a moment. A human being has approximately 86 billion neurons, nearly all of them gathered in the skull and spinal cord, a centralized system that sends and receives long cables of command and sensation throughout the body. The muscles of a human hand do not decide anything, they wait. The decision travels down from above, arrives in milliseconds, and the hand moves because the brain told it to. The hand itself is passive in this arrangement, a receiver and executor carrying no intelligence of its own. The octopus arm is not a hand, it is something closer to a collaborator. When an octopus reaches into a crevice to investigate whether something edible lies inside, the arm does not simply transmit the sensation of what it finds back to the brain and wait for an answer. The arm itself evaluates what it has touched. The neurons in the arm cord process the mechanical properties of the object, its hardness, its surface texture, its resistance, and generate initial motor responses on the basis of that local processing alone. The brain may receive a summary, may issue broad directives, but the arm is already handling the details. The investigation is already underway before the brain has finished forming a response. The evidence for this is not theoretical. A severed octopus arm, detached entirely from the animal, no longer receiving any signal from any central structure, will continue to respond to stimuli for up to an hour after detachment. Place a piece of food in contact with a severed arm on a laboratory surface, and the arm will curl around it. It will draw the food toward the location where the mouth would be if the mouth were still there. The arm is performing a complete feeding behavior, coordinated and purposeful in its mechanics, in the total absence of any brain. It has no way of delivering food to anything. The mouth it is reaching for is gone, and yet the behavior runs to completion because the nervous tissue in the arm still contains enough of the program, enough of the architecture of coordinated response to carry it through. This is not a reflex in the ordinary sense. A reflex is a simple arc, a stimulus reaching a small cluster of neurons, those neurons triggering a fixed response, the whole thing over in a fraction of a second. What the severed arm performs is more complex, a coordinated multi-step motor sequence involving multiple suckers, multiple joints, a sustained curvature extending along most of the arm's length, all organized toward a behavioral goal. The arm is not twitching, it is reaching. The implication is quiet, but considerable. If behavior this organized can run in the complete absence of the brain, then the arm contains within its own axial nerve cord something that deserves to be called processing, genuine evaluation, and response generation distributed far from the center of the animal. The octopus is not a brain with 8 extensions. It is something more like 8 semi-autonomous agents coordinated by a central executive, the whole arrangement functioning as a single animal with a form of intelligence spread across an entire body, like weight distributed evenly across a wide base. 8 arms lying loosely across a rocky seafloor, each one in contact with a different patch of stone, each one receiving a different set of chemical and mechanical signals from the substrate beneath it, each one doing its own quiet work of evaluation. Imagine 8 conversations happening simultaneously, none of them waiting for the others to finish before continuing. The rock is cold here, the rock is rough there. There is a faint trace of something biological at the tip of the third arm, something that may be worth investigating. All of this is being processed in parallel. Right now, the brain is aware of the broad shape of the situation. The arms are already in the details. Across the surface of the skin, something else is happening. Cells called chromatophores are holding their current state, expanded, contracted, half deployed, maintaining a pattern against the rock that began when the animal settled here and has been actively sustained at the cellular level ever since. Each chromatophore is a small elastic sac filled with pigment, brown, orange, red, black in different cells. Each sac is surrounded by radial muscle fibers that pull it outward when they contract, expanding the cell from a tiny contracted dot to a wide disc in a fraction of a second, revealing color. When the muscles relax, the sac retracts back to its pinpoint. The expansion and contraction of a single chromatophore from fully contracted to fully open can take as little as a third of a second. Across a body surface covered in millions of these cells, the combinations of expansion and contraction produce the patterns that the eye reads as camouflage, display, or signal. What makes the chromatophore system remarkable is not the speed, though the speed is considerable. What makes it remarkable is its directness. Each chromatophore is wired to its own motor neuron. There is no intermediate layer of computation between the nerve signal and the cell. The signal arrives, the muscles contract, the cell opens. This is one of the most direct neural control systems for rapid color change found anywhere in the animal kingdom, each unit of color production under its own individual neural command. The entire skin surface available for coordination at a resolution that is, in principle, as fine as the individual cell. A single chromatophore, seen in close focus under the right light, expands from a barely visible point of dark brown to a wide disk of deep amber in the time it takes to blink. One cell among millions, and around it, in the same fraction of a second, its neighbors are doing the same, or the opposite, or something in between, the whole surface rewriting itself, not in one instant, but in a wave, a flowing change that begins at one edge of the body and crosses to the other in a fraction of a second, the color and pattern of the animal shifting as a single continuous act. The skin is not simply a display screen. It is a sensory surface as well, responding to light, to touch, to the chemical character of the water moving over it, and the signals passing through it are traveling in both directions at once. The chromatophores are expressing something. The mechanoreceptors and chemoreceptors embedded in the same tissue are receiving something. The distinction between output and input, between speaking and listening, is harder to draw cleanly here than it is in the nervous systems of animals, with more clearly segregated sensory and motor functions. In the octopus, expression and perception are interwoven in the same tissue, wired through the same dense local networks, running simultaneously across the same surface. The arm nerve cords, the chromatophore system, the chemosensory suckers, these are all peripheral. They are the distributed parts, the parts spread across the body's geography. At the center of the animal, tucked inside the mantle, and threaded around the esophagus, in a configuration that is itself unusual, the esophagus passes directly through the brain mass, which means that anything the octopus swallows must pass through the center of its own central nervous system, sits the brain proper. It is a compact structure by vertebrate standards, small enough that its individual lobes can be distinguished without magnification, divided into distinct functional regions, each dedicated to different aspects of neural processing, each connected to the others and to the peripheral nervous system through a network of pathways that neuroscientists spent decades mapping one region at a time. One of the researchers who spent the most sustained and careful attention on that mapping was a British neurobiologist named J.Z. Young, who worked across the middle decades of the 20th century and returned to the octopus nervous system again and again over the course of his career. Young's work established many of the foundational maps of octopus brain structure, tracing the connections between lobes, identifying which regions were involved in which classes of behavior, and through careful and systematic lesion studies, in which specific brain regions were removed and the resulting behavioral changes observed, beginning to assign functions to structures. One structure that emerged from Young's lesion studies as particularly important was a region he called the vertical lobe. It sits in the supraesophageal brain mass, the upper portion of the brain, above the esophagus, as a small, dense, organized structure, tightly packed with neural connections, running in a characteristic pattern that Young and later researchers found shared features with structures in vertebrate brains associated with memory and learning. In itself, the vertical lobe is not large. Pressed flat, it might cover the face of a small coin. Its mass in a typical adult octopus is not much more than a small peppercorn in weight. What Young's lesion experiments revealed was specific and instructive. When the vertical lobe was removed, octopuses could still perform tasks they had learned before the surgery. Short-term performance on learned behaviors was largely intact. What failed was long-term retention. An octopus with a removed vertical lobe could learn a discrimination task, learn to choose one object over another on the basis of the reward it produced, but when tested days or weeks later, the learning had not persisted. The same task had to be relearned from the beginning, as though the first training had never happened. The memory had not consolidated. Something that should have settled into long-term storage had instead faded, leaving no trace. This placed the vertical lobe specifically in the business of consolidation, the process by which an experience is converted from a transient neural state into something more durable, something that can be retrieved later when the original experience is long gone. Not the initial learning, not the short-term performance, the specific step of taking what happened today and making it into something that will still be known tomorrow. The vertical lobe does not do this alone. Consolidation is a process that unfolds over time, drawing on multiple structures, running partly during quiet periods and partly during sleep, involving the strengthening of some synaptic connections and the pruning of others, the gradual reorganization of a temporary trace into a more stable pattern. But within the octopus brain, the vertical lobe appears to be the structure whose presence is necessary for that process to complete. Without it, experience passes through the animal and leaves nothing behind. There is something worth holding quietly in that finding. The octopus has 500 million neurons, most of them in the arms, the whole system capable of producing extraordinarily sophisticated local behavior without any reference to the brain at all. And yet the part of the brain that makes learning permanent, the small dense structure that takes the day's experience and converts it into memory, is a structure the size of a peppercorn in an animal whose intelligence is otherwise distributed so widely that the concept of a central location barely applies. The most dispersed nervous system among the complex invertebrates, and at its center, a tiny archive quietly doing the work of preservation. The neuron counts, when laid out with some care, begin to suggest the shape of what this kind of mind might feel like from the inside, if that question has any meaning here, which may itself be one of the quietest open questions in the biology of animal cognition. 350 million neurons distributed through the arms, each arm a corridor in a library that runs along the seafloor, reading the world through its suckers as it moves. 50 million more gathered in the two large optic lobes, one on each side of the brain, devoted entirely to the processing of visual information, a massive investment in sight, consistent with the octopus's extraordinary visual acuity and the sophistication of its visually guided behaviors. And then the remainder, the smaller central archive, in the brain lobes responsible for integration and higher order processing, coordinating the reports from the arms, the images from the eyes, the signals from the skin, and weaving them into the continuous state that guides the animal through its day. The proportion is striking. An animal with vertebrate-scale neuron counts has allocated the bulk of its neural resources not to a central processor, but to the periphery, to the arms that touch the world directly. The hierarchy implied by vertebrate brain organization, sense, transmit, process centrally, respond, is replaced here by something more lateral, more distributed, more immediate. The processing happens close to the source. The arms do not send raw data to a distant center for interpretation. They interpret locally, respond locally, and report the outcome. The seafloor at night, cold and dark, the surface of the rock running beneath an arm that has been resting here for several minutes. Across those minutes, the arm has sampled the rock at dozens of points along its length. It has detected the faint presence of a prey organism somewhere in the crevice beneath it, a chemical trace in the water that has seeped up through the gap between two stones. The arm has not waited for permission to investigate. It has already begun to angle toward the gap, already begun to adjust the pressure and orientation of the suckers nearest the trace. The action emerging from the arm's own processing before any signal from the brain has decided that investigation is called for. The brain, When the signal from the arm's nerve cord reaches it, we'll catch up. It will incorporate what the arm has already found into the animal's broader picture of its situation. It may issue a directive, or it may simply ratify what the arm has already begun. This is a nervous system that runs differently from the nervous systems of animals whose architectures most people have grown up learning about. Not faster or slower, not better or worse in any simple sense. Different in kind, organized around a different principle of where computation should happen and how far it should be distributed from the site of sensation and action. The brain of a fish, a bird, a mammal pulls sensation in and pushes response out through a hierarchy whose center is the brain itself. The octopus keeps more of the work at the edge, close to the world, distributed through the 8 corridors of the arms, with the central brain setting context and holding the long history of what has been learned. The arm on the rock stirs slightly. The suckers at its tip have found the edge of the crevice. In the darkness at the bottom of a shallow coastal sea, in the cold and quiet of whatever hour this is, the arm begins to move. Along its entire length, the nerve cord is active. Signals are passing in many directions at once, from sucker to cord, from cord to sucker, up toward the brain, back down again. At the skin surface, chromatophores are responding to some shift in the visual field, a change in the light falling on the rock nearby, a shadow that may indicate movement. Some expand, others retract. The pattern on the skin shifts slightly, settling into something slightly darker at the edges, slightly more mottled at the center, matching the rock more precisely than it did a moment ago. None of this required conscious direction. The cells responded to the signals they received through the motor neurons wired directly to them, and the pattern adjusted accordingly. The arm continues its investigation. In the brain above, in the small lobes of the supraesophageal mass, the vertical lobe is receiving the current moment, receiving the sensory summary of what the arm has found, what the eye to the left has seen, what the skin has reported about the quality of the light. And somewhere in that dense peppercorn-sized structure, the current experience is being held alongside the record of previous experiences in this same kind of situation, the trace of what happened before, of crevices that yielded prey and crevices that did not, of the particular chemical signature of a crab versus a worm, of the texture of a rock that turned out to be something else, is being compared, quietly and without any urgency, to what is happening now. This is how the octopus knows what it knows, not through language, not through anything resembling deliberate recall, but through the retrieval of consolidated associations, sensory patterns laid down by previous encounters, preserved through the specific work of consolidation that the vertical lobe makes possible, available now, when the present moment matches them closely enough to trigger their reactivation. The past reaches into the present through this structure. Experience accumulated over weeks of hunting along this section of seafloor shapes how the arm responds now, how long the investigation continues, how much pressure the suckers apply to the edge of the crevice. A small structure doing something vast. Not vast in the dramatic sense. There is no drama here, only the quiet and continuous operation of a biological system that has been running in various forms for hundreds of millions of years, refined by the long, slow process of variation and survival until it works with the precision and efficiency that allows this animal to live, hunt, learn, and persist in a world that is almost entirely cold and dark and full of things that would eat it. The vertical lobe does not know it is preserving anything. The arm does not know it is processing. The chromatophores do not know they are expressing a pattern. And yet, across the whole distributed system, something is happening that no single part of it could do alone. The arm lies against the rock. The suckers are reading the stone. The nerve cord is already at work. In the mantle above, the brain is receiving, holding, comparing, and in the dense quiet of the vertical lobe, the experience of this moment is being pressed into a form that will survive the night. Somewhere in the architecture of that small structure, the geometry of today is slowly becoming the knowledge of tomorrow, one narrow increment of consolidation at a time, the dim present reaching forward into the persistence of memory, the world outside the animal passing through a window, no larger than a peppercorn, and leaving something that will still be there when the light returns. The arm is still, the rock is cold, the process continues, unhurried and complete. A glass jar sits on the floor of an aquarium tank. It is not large, no wider than a fist, no taller than a hand's length, but sealed at the top with a lid screwed tight, and inside it, visible through the curved glass wall, is a piece of food. The jar presents a specific kind of problem. It cannot be bitten through. It cannot be pulled apart. The food inside it is real and close, and entirely inaccessible by any direct means, and the only solution available requires understanding, at some level, that the lid and the body of the jar are two separate things, joined by a relationship that can be undone. An octopus approaching this jar does not rush. It arrives the way octopuses arrive at most things, slowly, with several arms extended ahead of it, the suckers along each arm already reading the glass surface for texture and temperature, and the faint chemical trace of what is sealed inside. Two arms reach the jar first. They move across it without apparent purpose for a moment, feeling along the cylindrical surface, finding the seam where the lid meets the thread of the neck. Then something changes. The arms that were exploring begin to grip. The suckers lock, and the animal applies, through the coordinated tension of muscles distributed along two arm lengths, a slow rotational force to the lid. It does not open on the first attempt. It rarely does, the first time. What happens across repeated trials is quieter and more interesting than any single success. The time between approaching the jar and opening it grows shorter, not because the octopus grows stronger, but because something in the process has been retained and simplified. The unnecessary exploratory passes reduced, the grip found more quickly, the direction of rotation applied with less apparent searching. The jar that took several minutes to open the first time takes under a minute by the third or fourth encounter. The knowledge of how to do it has settled somewhere in the animal and remains available, and this has been documented at marine biology laboratories across Europe and at institutions including the Seattle Aquarium, each record another entry in the quiet case that what the octopus is doing when it opens a jar is not luck and not reflex, but something that belongs to the category of learning. The specific challenge of a jar lid is worth pausing on. To unscrew it, the octopus must apply torque, rotational force, which requires holding the body of the jar stable while rotating the lid relative to it. These are two opposing actions performed simultaneously on a single object. The animal has no rigid skeleton, no lever system of bone and joint to brace against. Everything it does in soft tissue. The coordination required, some arms gripping and holding, others applying directed rotation, the body positioned to provide counterforce, draws on the distributed intelligence of the arm system, each arm receiving and processing its own sensory stream, each contributing to an action that, from the outside, looks purposeful and whole. The food inside the jar is real, the problem is real, and the solution repeated and refined across trials, is real in the way that learning is real, not as a single bright event, but as a slow accumulation, a trace that deepens each time it is used. Somewhere in that jar opening, in the shortening interval between arrival and success, there is a record being kept, not in ink or stone, in the altered strength of connections between neurons, in the geometry of a nervous system that has encountered this problem before and carries the memory of it forward through time. There is another kind of knowing that does not come from doing the thing oneself. An octopus on one side of a transparent barrier watches another octopus on the other side. The second octopus has been trained, through patient repetition with food rewards, to approach a particular object rather than another. It does so now, moving toward the correct target with the ease of something long practiced. The watching octopus cannot touch either object. It cannot taste or smell the reward. It can only see the other animal's choice and the outcome of that choice through a wall of clear glass that transmits light and image, but nothing else. When the barrier is removed and the watching octopus is given its own access to the objects, it approaches the correct one, the one it saw chosen, at rates significantly higher than octopuses that had not watched. It did not learn through practice. It did not learn through error and correction. It learned, if that word can be used carefully here, by watching the correct object was marked for it, not by its own experience, but by the experience it witnessed in another. This is notable for several reasons, and the quietest of them is this. Octopuses are largely solitary animals. They do not form groups. They do not coordinate hunts or share territories in ways that would obviously benefit from social learning. And yet the capacity appears to be present, the nervous system wired, somehow, to extract useful information from the behavior of a conspecific observed at a distance. What shapes that capacity in an animal with so little apparent social life is an open question. That the capacity exists has been demonstrated. The two facts do not fully resolve against each other, and they do not need to. The glass barrier stands between two animals. One moves, the other watches. And what passes between them, through the transparent wall, along the axis of vision, without touch or sound, or shared space, is enough to change what the watcher reaches for when the barrier is gone. Not every action an octopus takes is aimed at food. Not every movement ends in eating. This is worth stating plainly, because it is easy to describe animal behavior entirely in terms of the resources it secures, the prey caught, the shelter claimed, the predator escaped, and to miss what does not fit that frame. Jennifer Mather and her colleagues, watching octopuses in controlled tank environments, documented something that takes a moment to describe without overstating it. An octopus would take a small buoyant object, a plastic bottle, a floating pill container, and propel it toward a water jet in the tank. The current would carry the object away. The octopus would retrieve it and propel it toward the current again. This happened not once or twice, but in sequences cycling up to 20 times in a single session. The animal was not hungry. The object was not food. No reward was associated with it. The action produced nothing except its own repetition. Several criteria exist in ethology for identifying behavior as play. The behavior should occur in the absence of immediate need, when the animal is not hungry, not threatened, not in search of a mate. It should be repeated and apparently voluntary. It should be structurally similar to functional behavior, but detached from the function. It should show variation and some sensitivity to novelty. By these measures, what Mather's octopuses were doing with the floating objects fits multiple criteria for play. Not conclusively, because animal behavior is rarely conclusive, and because play is a category that resists clean edges, but the behavior was there, cycling and purposeless and repeated, and it was seen, and it was recorded, and it asks something of anyone who looks at it. A small plastic bottle drifts back on the current toward an animal that has already released it once. The animal releases it again. It drifts back. The sequence has no ending except the one the octopus eventually makes by losing interest and moving on. There is no food at the end of it. There is no safety. There is only the motion repeated and the animal choosing to repeat it, and the strange fact of that choice in a creature whose nervous system is organized so differently from the vertebrate systems in which play was first described and studied. The bottle drifts, the current returns it, and for a time, apparently, that is enough. Memory in the octopus is not a single uniform thing stored in a single place. It has textures and time scales, and the architecture that supports it is distributed across brain regions that serve different aspects of what it means to know something across time. There is a kind of knowing that lasts only a moment, the position of a prey animal, the texture of the surface directly underfoot, the temperature of the water passing over the skin. This kind of knowing does its work and dissolves. It does not need to last. And there is another kind that persists, the association between a particular surface texture and a food reward, retained weeks after the last encounter with it, available to guide a choice in a new context far removed from the original learning event. Octopuses trained to discriminate between objects by touch, to associate a rough surfaced shape with food, and to ignore a smooth one, for example, retain that discrimination for at minimum several weeks without any intervening reminder. When the rough object is offered again after that interval, the arm reaches for it with the same directness as the arm that learned to reach for it in the first place. The memory held. It held through whatever the octopus experienced in the weeks between, through meals and movements and the daily business of surviving, and it was still there when the test came again. The structure that matters for this long-term retention is small and dense, located in the upper region of the octopus's central brain mass. It is called the vertical lobe, and its role in memory has been established through careful lesion work, experiments in which specific damage to the structure produced specific and revealing deficits. An octopus with an intact vertical lobe can learn a tactile discrimination task and retain it. An octopus with damage to the vertical lobe can learn the same task in the short term, can show it has registered the association, but the retention does not hold. The experience does not become the lasting knowledge it should. What was learned today is gone tomorrow. The vertical lobe is not involved in the moment of learning. It is involved in the conversion of what was learned into something that lasts. The distinction matters because it means the octopus brain separates these two functions, the acquisition and the consolidation, the way that vertebrate brains do, though through an architecture that evolved entirely independently along a lineage that diverged from the vertebrate line more than 500 million years ago. A rough surface, felt once by the suckers of an arm associated with food, filed away. Weeks later, in a different context, that same surface is offered, and something in the animal already knows what to reach for. The knowing is older than the moment. It came from a moment that is now weeks in the past, transformed somewhere in the dense tissue of the vertical lobe into a permanent availability, a shape in the architecture of the nervous system that is, in whatever sense matters, memory. What the octopus remembers, it does not only know, it carries, and what it carries shapes what it does, quietly, without any visible sign of recollection, just the arm reaching without hesitation for the rough surface, because in the deep structure of the nervous system, that choice has already been made. Place two octopuses of the same species in separate but identical tanks. Give each of them the same novel object, something small and unfamiliar, placed at the center of the space. Watch what happens. One octopus will extend an arm toward the object within seconds of noticing it. The arm reaches without apparent hesitation, the suckers making contact, the animal's body already beginning to move toward whatever it has found. There is no visible deliberation. The approach is quick and continuous, as though the decision, if it can be called that, was made before the movement began. The other octopus withdraws. It moves to the back of its den, if it has one, and stays there. Its arms are pulled close. Its body is compressed slightly, the way octopus bodies compress when the animal is alert, but still, from that distance, it watches the object. Not briefly for a long time, long enough that a human observer might think it has lost interest, or that nothing will happen. And then, eventually, slowly, it extends one arm toward the object from the maximum safe distance, touches it lightly, and withdraws again. The approach, when it finally comes, is incremental and interrupted, each advance followed by a pause, each touch brief. These two animals are responding to the same object, in the same conditions, at the same moment in their captive lives. They are the same species. Their nervous systems are built on the same plan, and yet the behavior is so different that watching them, it is difficult not to feel that they are expressing something intrinsic to who each one is, something stable and characteristic that belongs to that individual animal and not to the species in general. This is not just the impression of observers who spend time with octopuses. Though such observers report it consistently, it has been measured. Individual octopuses show consistent, repeatable behavioral profiles across different contexts and across time, profiles that include axes like boldness versus shyness and activity versus inactivity that do not change with a single altered condition and that persist across weeks of observation. An octopus that approaches novel objects quickly in 1 week tends to approach them quickly in the following week. An octopus that withdraws and watches tends, when tested again, to withdraw and watch. The pattern holds. In the formal language of animal behavior research, this is called a behavioral syndrome, a consistent covariation of traits across contexts that is stable enough to constitute something like a temperament. The word carries no implication of inner life, no claim about what an octopus experiences or whether it experiences anything at all. It describes something observable, a pattern, a repeatable difference between individuals that belongs to each one, the way that coloring belongs to an individual, or the particular shape of its arm musculature. Not chosen, but characteristic. There is something in this that resists the expectation that intelligence works uniformly within a species, that all octopuses face the novel object the same way, or that any given octopus would behave identically in every context if only the conditions were right. They do not. The bold one is bold in the morning, and bold a week later, and bold in a new tank. The cautious one watches longer. Always, the difference is stable. It is their own. Two octopuses in the same genus, the same species, the same environmental conditions, the same developmental history of captivity. One reaches, one watches. Each one is being, in whatever sense the word applies, exactly who it has always been. There are questions that gather naturally around what octopus cognition is at its foundation. What does it mean to learn something when the learner has no fixed architecture of bone, No centralized control, 8 semi-autonomous arms, each conducting their own sensory conversation with the environment. What does problem solving look like when 2/3 of the neurons available for solving the problem are not located in the brain at all, but in the arms that are doing the solving? An octopus working on a jar lid is not simply executing a centrally planned motor program. The arms contribute. Each arm carries sensory information from the suckers, from the skin, from the proprioceptive signals that track the arm's own position and tension, and processes a portion of that information locally before any of it reaches the central brain. The rotation of the lid is felt in the arms before it is represented, if it is represented in any central structure. The feedback that guides the continuing force and direction of the movement comes partly from the arm itself, running on its own processing power, adjusting as it goes. This does not mean the central brain is uninvolved. The decision to approach the jar, the learned knowledge that turning this particular structure produces food, the allocation of attention across the multiple arms engaged in the task, these are coordinated at a level above any single arm. The arm and the brain work together, but not in the way that a hand and a motor cortex work together, with clear hierarchy and a single center of command. The relationship is more distributed, more mutual, less like a commander and a soldier, and more like 8 partially independent agents operating under a loose common understanding, each contributing something the others cannot provide. What emerges from this arrangement, in the jar and at the barrier, and with the floating bottle, and across the weeks of a remembered texture, is something that looks like intelligence. It looks like intelligence because it solves problems that require retaining information across time, adjusting behavior based on observation, and acting flexibly in the face of a novel situation. Whether it is intelligence in the same sense that the word is used for a primate or a corvid, or any other animal studied in the cognition literature, is a question that remains carefully open. The tools and the architecture are different. The results in certain constrained experimental contexts are comparable. The comparison is informative without being complete. The octopus does not solve a jar because it resembles a primate solving a puzzle box. It solves a jar because it is what it is, an animal with 500 million neurons arranged in a way that has no close parallel in the history of animal life, approaching a specific problem in the specific way that its specific body makes possible. The intelligence is real. The terms for it are still being found. A jar sealed, a barrier clear, a bottle floating, a texture remembered. These are small things. A jar that fits in a human hand, a piece of food inside it, a transparent wall between 2 animals in a laboratory tank. They are not the stuff of ocean depths or geological time. They are the ordinary materials of a controlled experiment, chosen because they are simple enough to study, constrained enough to measure, simple enough to compare across individuals and trials and laboratories. And yet through these small things, something becomes visible that would otherwise remain hidden in the architecture, the octopus reaching for the correct object, not because anything forced it, but because it watched. The octopus opening the jar faster on the 3rd try than on the 1st, not because it grew stronger, but because it learned. The octopus propelling the bottle toward the current and watching it return, for no reason that can be expressed in terms of survival, but because it is doing something that looks, in its quiet and purposeless repetition, like play. These are not the same kind of observation. Learning and play and social attention are different phenomena with different neural substrates and different evolutionary histories. They are mentioned together here not because they collapse into a single category, but because they occur in the same animal in this cephalopod, with this nervous system, in this body made of soft tissue and distributed neurons and arms that think for themselves, and their coexistence in a single organism is part of what makes the organism worth paying attention to. The floor of an aquarium tank is lit from above, the water carrying the light downward in slow columns that shift and scatter as the surface moves. An octopus sits at the center of the tank, two arms extended, the rest gathered loosely beneath it, its skin pale and still. The jar is 10 inches away. It has not moved toward it yet. What the octopus knows, or has known, or will know, what the vertical lobe holds in its dense tissue from one day to the next, what the arms carry in their own local processing, what the nervous system as a whole retains and uses across weeks and trials and changing contexts, none of this is visible from outside. None of it announces itself. The animal sits, and the jar sits, and the light moves above them both, or whatever is happening in the tissue of the animal's body is happening in the quiet interior of a kind of mind that took a completely different path to arrive at something that can learn from a jar, remember a texture, watch a conspecific and be changed by watching. The octopus moves. Two arms extend. The suckers find the glass. Individual octopuses do not merely vary from one another in boldness or caution on a single occasion. The variation is consistent, and what makes it consistent is that it expresses something about the organization of that individual nervous system, some stable pattern in the way signals are weighted, in the thresholds at which approach or withdrawal is triggered, in the particular balance of competing tendencies that constitutes the behavioral architecture of that one animal. This is not unusual across the animal kingdom. Birds show consistent individual differences. Fish do. Insects do. In some studied species, personality, or something like it, stable individual variation in behavior appears across a wide range of taxa, and its presence in octopuses is continuous with its presence elsewhere, not a special wonder belonging only to this group. What is notable, given the architecture of the octopus nervous system, is where that consistency must be maintained. If most of the neurons in an octopus body are in the arms, and each arm has substantial local processing autonomy, then the individual behavioral style of the whole animal is not simply an output of a single centralized personality module. It must be expressed consistently across a body that is, at the level of hardware, distributed and semi-autonomous. The bold octopus is bold in all 8 arms simultaneously, even though those arms can function independently, even though they do not share a single central decision-making unit in the way a vertebrate brain centralizes voluntary action. The consistency percolates through the architecture, stable at the level of the whole animal, despite being grounded in a system that is not organized around a single center. How that works, how individual variation is maintained coherently across a distributed system, is one of the genuinely open questions in cephalopod biology. The observation that it does work, that individual octopuses are consistently themselves across contexts and time, is clear. The mechanism that produces and maintains that consistency in a body organized the way an octopus body is organized is less clear, and the picture that will eventually account for it fully does not yet exist. What is known is enough to hold on to. The cautious octopus withdraws, and watches and will withdraw, and watch again tomorrow. The bold one reaches and will reach again. Neither of them is performing their behavioral profile. Neither of them is aware of it as a pattern. They are simply being what the organization of their nervous system, built through development and perhaps refined through experience, makes them most inclined to be. There is something restful in the consistency of individual creatures, in the knowledge that within each one certain tendencies hold, that the animal at the back of the den today is not randomly cautious, but characteristically cautious, that this is its particular way of meeting the world. The world contains many kinds of minds, organized in many ways, and one of the quieter facts about those minds is that each of them, down to the level of an individual octopus in a laboratory tank on a slow afternoon, carries its own character, persistent, and its own. The last light in a tank moves slowly across the substrate. An octopus has returned to its den, two arms trailing outside it, relaxed. The jar is open, the food is gone. Whatever the animal did to get it, the grip, the rotation, the coordinated force distributed through arms that each contributed something, has been done, and the record of having done it is already settling into the deep architecture of the nervous system, folding itself into the connections that will make the next attempt shorter. In the weeks ahead, if the jar is offered again, the animal will be ready for it, not because it will remember in any way it can express or demonstrate, but because the memory will be functional, present in the altered geometry of the nervous system, available to guide the arms without the animal needing to search for it. The rough texture of the jar's lid, the direction of rotation, the particular bracing of the arms that hold the body and the jar steady while two others turn, all of it filed, refined, available. Memory, in this sense, is not a record kept in a separate archive. It is built into the body, into the weights and thresholds of the system that learned it, distributed through the arms and the central lobes, in proportion to the roles they played in the original action. The arm that turned the lid retains something of the turning. The brain that recognized the jar retains something of the recognition. The vertical lobe, dense and small, holds the consolidation that makes the whole thing last. This is not uniquely the octopus's way of holding memory. Distributed encoding of motor and sensory experience is not exclusive to cephalopods, but in the octopus, the distribution is more radical, the autonomy of the components greater, the architecture further from the vertebrate model against which most of this has been measured. To understand how the octopus learns and remembers and plays and recognizes a conspecific's choice and expresses in its own consistent way its own particular character, all of this requires understanding that the animal carrying out these behaviors is built on a plan that has no close structural relative in the vertebrate world. It is not a smaller or simpler version of vertebrate cognition. It is not a less developed form of anything. It is the expression of 500 million neurons, arranged in a way that evolution arrived at independently, in a lineage of animals that diverged from the vertebrate line in the ancient sea, and that has had since that divergence an enormous amount of time to develop its own solutions to the problems of surviving, feeding, hiding, learning, and being alive. The octopus opens the jar. The octopus watches through the glass. The octopus releases the bottle into the current and lets it return. The octopus holds, in its dense vertical lobe, the memory of a rough surface felt weeks ago. The octopus at the back of its den waits, and eventually extends one arm toward the unfamiliar object and touches it, and that is exactly who it is. Each of these things is a small thing. None of them requires a vast stage or a dramatic frame. They happen in tanks of lit water, in quiet laboratories, in the patient hours of researchers watching and recording, in the interior of an animal sitting at rest on a rocky substrate while its arms read the world around it and its nervous system does what it does, unhurried, in the dark. Intelligence, in the octopus, does not announce itself. It is present in the shortening interval between problem and solution. It is present in the choice that follows watching, It is present in the reach toward the rough surface after 3 weeks of not encountering it. It is present in the reliable withdrawal of the cautious animal and the reliable advance of the bold one, each one meeting the world in the way that is, for that individual, simply how meeting the world is done. Different individuals, different approaches. One kind of nervous system, radically distributed, built from soft tissue in slow time, arriving at something that in certain important and measurable ways is a form of knowing, and in each individual a form of being that belongs to no other group of animals alive. The tank is quiet now. The arms are still. Whatever is being held in the tissue of the nervous system, in the connections altered by today's experience, in the slow chemical consolidation that will, across the hours ahead, deepen what was learned into something that will still be available weeks from now. All of that is invisible, interior, proceeding without any outward sign, and the octopus rests in its den, arms gathered close, skin pale and even, carrying in the architecture of its body an account of the world as it has met it, a particular individual account, shaped by what this specific animal has encountered and retained, different from the account carried by any other octopus in the ocean. Each one is its own kind of knowing, particular and persistent, quiet as the water around it, and complete. At 600 meters below the surface of the ocean, the light does not fail all at once. It thins. It drains away in increments, each 100 meters removing another layer of color, another degree of brightness, until what remains is not darkness, exactly, but something closer to the memory of light, a faint, directionless blue-gray that carries no warmth and comes from no particular place. The water here is cold, 4 degrees Celsius, perhaps 5, a temperature that has not changed in years and will not change tomorrow, the same cold pressing in from every direction with a weight of 60 atmospheres bearing down on every surface. This is the mesopelagic zone, the ocean's middle layer, and it is one of the largest inhabited spaces on the planet, a volume of cold, dim water stretching from roughly 600 feet below the surface to nearly a mile down, encircling the entire Earth, and almost entirely beyond the reach of any eye that has not adapted over a long time to find what light is left. There is still some light here of that faint blue quality that filters down from above. It is not enough to read by, not enough to navigate a room, not enough to distinguish the color of a hand held before the face, but to an eye shaped by many generations of life, at this depth, it is enough. It is enough to perceive movement 30 meters away, enough to catch the slow pulse of a bioluminescent body drifting past in the water column, a cold blue-green flash lasting less than a second. Enough, if an animal is patient and still enough to wait, to understand the difference between food and danger. The mesopelagic zone is not empty. It is among the most densely populated regions of the ocean, filled with animals that have learned to live inside the threshold between light and dark, migrating upward each night into richer, shallower water, sinking again each dawn to hide in the dimness below. The pressure at this depth is not something the body registers gradually. It is a constant. Every organism in this zone carries it the way a surface animal carries the weight of the atmosphere, as a condition of being, unremarkable, not perceived because it has never not been present. At 600 meters, the water above presses down at 60 times the force it exerts at the surface. At 1,000 meters, that figure approaches 100 atmospheres. These are not numbers that the body can translate into familiar sensation. The closest comparison is this: 100 atmospheres is roughly the pressure one would experience pressed beneath a column of water as wide as a 10-story building is tall, on every square inch of skin, from every direction at once, without interruption. Surface animals placed here would not survive the transit. Their cellular membranes, built to function under ordinary atmospheric pressure, would find their fluidity disrupted, their protein structures distorted, their metabolic chemistry altered in ways incompatible with life. The animals of the mesopelagic zone do not find this remarkable. They were built for it, across long generations of selection. At the molecular level, the cold presents its own constraint. At 4 degrees Celsius, chemical reactions slow. The enzymes that carry out the chemistry of life, the proteins that fold and unfold millions of times per second to catalyze the reactions that produce energy and build tissue, do not behave at near freezing temperatures the way they behave in the warmth of a summer tide pool. Enzymes are shaped structures, and their shape determines their function. Cold stiffens them. At temperatures approaching freezing, a surface-adapted enzyme may simply cease to work, its molecular motions too sluggish, its catalytic geometry too constrained by cold to complete its task. Deep-sea animals carry different versions of the same enzymes. Their proteins have been altered through long inheritance to remain flexible and functional at 2 or 3 degrees Celsius to catalyze their reactions efficiently in water that is barely above the temperature of melting ice. These are cold-adapted variants, and they come with a trade-off. They are unstable at warmer temperatures, would lose their shape and their function within seconds in the water of a shallow summer sea. The deep is their only home. Cold-adapted enzymes allow survival, but they do not accelerate it. Chemical reactions in cold water are inherently slower than the same reactions in warm water, and no amount of molecular adaptation fully overcomes this. Growth is slow in the deep ocean. Reproduction is slow. The metabolic pace of life here is measured and unhurried in a way that surface life, burning fast on abundant light and warmth, simply is not. A deep-sea cephalopod growing toward maturity does so across a span of time that a warm water counterpart might spend and finish in a fraction of the duration. Food is scarcer here. Energy must be spent carefully, only on what is necessary. The body maintains itself at the lowest cost that allows it to continue, and it continues for a long time, and it is patient in a way that the biology of warmer, shallower places does not require. In the permanent cold of the mesopelagic zone, where food arrives irregularly and the dark offers little navigation, the senses that matter most are not always the ones that serve well in light. Chemoreception, the detection of dissolved molecules in the surrounding water, is acute in many deep-sea animals, a way of reading the water for traces of food or threat that requires no photons at all. Mechanoreception, the sense of pressure changes and water movement, provides information about the proximity and motion of nearby animals in a medium that conducts vibration well. And then there is light itself, reimagined not as something to see by, but as something to make. Bioluminescence is not rare in the deep ocean. It is in fact so common at mesopelagic depths that researchers who have sampled the zone broadly estimate that a large majority of the organisms living there produce light of some kind. This light is cold. It is not the byproduct of heat or combustion, but the result of chemistry, a molecule called luciferin reacting with oxygen in the presence of an enzyme called luciferase, releasing the energy of that reaction not as heat, but as a photon, 1 photon per reaction, accumulated across many reactions happening simultaneously in specialized cells called photophores. This chemistry produces a glow visible in darkness, controlled by the animal, turned on and off as needed, shaped by the structure of the photophore into a beam or a diffuse wash, or a pattern of discrete points. The firefly squid is a small animal, no longer than 3 inches from mantle tip to arm end. It lives in the waters of the western Pacific, descending into the mesopelagic zone through the day and rising toward the surface at night, and it carries photophores across much of its body, on the arms, on the mantle, around the eyes, along the ventral surface. These are not simple lights. They are structured, individually controlled, capable of producing subtle variations in output. The light the firefly squid produces serves multiple documented purposes. On the ventral surface, the photophores produce a faint, diffuse glow directed downward, calibrated to match the residual downwelling light from above. This is counter-illumination. Any predator below the squid, looking upward, would see against the dim background of filtered sunlight a dark silhouette, the shadow of the squid's body, interrupting the light. By producing light from its own underside at exactly the right intensity to match the background, the squid erases that silhouette. It becomes, from below, part of the water. The predator finds nothing where the squid is. This is a precise calibration. Too bright, and the ventral surface shines as an obvious point in the dimness. Too faint, and the silhouette returns. The system that controls it must monitor the ambient light from above and adjust the photophore output in response continuously as the squid moves through layers of varying brightness. The animal manages this in real time without thought, through nervous pathways that connect the skin to the brain in a feedback loop operating across the animal's entire life. It is not intelligence of the kind that opens jars or solves novel problems. It is intelligence of a different order entirely, the accumulated solution to a problem that has existed for as long as there have been eyes in the dark ocean looking upward for food. Light in the deep ocean is not only a tool of concealment, it is a language, or something closer to a channel, a means of conveying information between animals in an environment where sound and chemistry travel. But light, generated by living bodies, is rare and therefore meaningful. Among mesopelagic squid, bioluminescent displays on the mantle and around the eyes have been observed in contexts suggesting communication between individuals, patterns that differ between circumstances, in ways that carry information about reproductive readiness, threat, or proximity. This is difficult to study in animals that live hundreds of meters down and die quickly when brought to the surface, and much of what the light means to the animals themselves remains unknown, but the structures exist. The photophores are present, precisely controlled, capable of producing varied outputs. The suggestion of a language written in cold light, in the dark water between animals that cannot easily find each other, is real, even where the full grammar of it has not yet been read. The descent continues below the mesopelagic zone, below 1,000 meters. The last trace of filtered sunlight is gone, not reduced, not faint, gone. There is no light here that does not come from a living body, and the living bodies are fewer and farther between. The water is colder still, closer to 1 degree above freezing, and the pressure has climbed past 100 atmospheres and continues to climb. Below 6,000 meters, in the trenches that plunge into the crust of the earth, the pressure exceeds 1,000 atmospheres. This is the Hadal zone, named for the underworld of Greek mythology, and it is among the least known inhabited spaces on the planet. The trenches are narrow relative to the ocean floor surrounding them, slots cut into the crust where tectonic plates meet, and one descends beneath the other, and they hold some of the most extreme conditions available to life on Earth. At 1,000 atmospheres, the biochemistry of life requires solutions more radical than cold-adapted enzymes alone. The membranes of cells, Thin sheets of lipid molecules that hold each cell's chemistry inside and regulate what passes in and out depend for their function on a specific physical property. They must be fluid, not liquid, not solid, but precisely between, a state that allows the proteins embedded in them to move, to rotate, to perform their functions as gates and channels and anchors. Pressure, like cold, disrupts this fluidity. It compresses lipid membranes toward a more ordered, less mobile state, and at Hadal pressures, the membranes of surface animals would stiffen past the point of function. Deep-sea organisms at these depths maintain membrane fluidity through the incorporation of unusual fatty acids, highly unsaturated chains that resist compression and remain fluid under pressures that would paralyze the membranes of shallower animals. These molecules do not occur in abundance in shallow water. They are produced by deep-sea organisms and by the microorganisms on which those organisms feed, and they accumulate in the tissues of hadal animals in concentrations reflecting the pressure of the world they live in. Cephalopod remains have been recovered from near 5,000 meters. Whether the animals were alive at those depths or sank there after death is not always clear, but the evidence of cephalopod presence in the deep ocean extends well below the mesopelagic zone, into the bathypelagic and upper abyssal waters, where pressure and cold reach extremes that surface life cannot tolerate. The octopus and squid that live at these depths are not the animals of the shallow reef. Their bodies are softer, their metabolisms more restrained, their reproductive strategies slower and more careful. They are adapted in ways invisible to the eye, in the molecular composition of their membranes, in the kinetics of their enzymes, in the structure of their proteins, to the specific and unrelenting pressures of the deep. There is one adaptation in the deep ocean that remains among the most striking in the animal kingdom, not because it is chemically complex, but because of its sheer physical scale. At the very deepest levels of the mesopelagic zone and below, in the permanent dark where bioluminescent flashes are the only light, vision still matters, predators still hunt, prey still hides. The detection of a faint flash of cold light 40 meters away can mean the difference between eating and going hungry, or between remaining unseen and becoming food. And for the largest hunters of the deep ocean, detecting that faint light demands an eye of corresponding size. The colossal squid is not the largest cephalopod by total length. Giant squid grow longer, their tentacles stretching to greater reach. But the colossal squid, which inhabits the deep southern ocean, carries something the giant squid does not, at least not quite to the same degree: its eyes. The eye of a colossal squid can reach 27 centimeters in diameter. That is wider than a large dinner plate. It is the largest eye of any living animal on Earth, and its size is not incidental. A large eye gathers more light than a small one, in the same way that a wide window lets in more of a dim day than a narrow slit. At the depths where the colossal squid lives, in the dark water beneath the Antarctic convergence, where bioluminescent flashes are faint and brief, a larger eye means more photons captured per flash, which means a brighter image from a dimmer source, which means a predator detected at greater distance. An eye 27 centimeters across in absolute darkness, waiting for the cold light of a bioluminescent body to cross its field of view, is an organ tuned to find what almost cannot be seen. An eye of this size could not function in the bright water of the surface ocean. The same structural features that make it so sensitive in darkness, the extreme size of the lens, the density of photoreceptors, the optical design of the whole, would produce an overloaded, non-functional image in bright light. The colossal squid's eye is built for one environment and one environment only. It is a deep water instrument, as specialized as any piece of apparatus designed for a single purpose, and it is extraordinary, not in the way that complicated things are extraordinary, but in the simple, direct way that a thing perfectly suited to its place always is. The eye does not move often. The colossal squid is a large, slow animal in cold, food-scarce water, and unnecessary movement is expensive. It holds position in the water column with minimal energy expenditure and waits. The eye is open. In absolute darkness, there is no reason to close it, no glare to shield against, no brightness that will damage the delicate photoreceptive tissue. It simply remains open in the cold and the pressure and the dark, and waits for something to pass through its field of view, the cold blue-green flash of a bioluminescent fish, perhaps, or the faint shadow of another large animal moving against the residual glow of distant photophores. The patience of this waiting is not a quality the animal possesses, the way a person might possess patience as a virtue. It is a metabolic fact. Cold, energy-scarce environments select for animals that spend little and wait long, because animals that move too much exhaust their reserves before food arrives. The stillness of the colossal squid in the deep ocean is not a choice. It is the shape of a life built to survive in a place where energy arrives slowly and is never wasted. The water around that waiting eye is not silent in the way that empty space is silent. Deep ocean water is not empty. It carries sound well, better, in some frequencies than air. And the creaks and groans of the Earth's crust, moving along the tectonic boundaries of the ocean floor, travel for thousands of miles in the deep sound channel. The clicks and sweeping calls of sperm whales hunting at depth, the same whales that prey on colossal squid, are audible across ocean basins. And beneath all of this, the simple physical sounds of water, pressure waves, currents, the occasional thud and drag of something large moving across the sediment of the abyssal plain 3 miles below. The abyssal plain itself lies below the Haddle Trenches in terms of depth, but it is far greater in area. It is the floor of the deep ocean, a vast, nearly flat expanse of soft sediment, clay, and silt, and the accumulated remains of organisms that lived in the water above over millions of years, sinking slowly to the bottom, building up in layers so gradual that in some places a centimeter of sediment represents 1,000 years of deposition. This is not a barren place, though it is sparse. Animals live here, adapted to cold and pressure and near total stillness, moving slowly across the sediment in search of food. Holotherians, sea cucumbers, drag themselves through the ooze, processing sediment for the organic particles buried within it. Small crustaceans, amphipods, move with quick purpose over and through the sediment. Polychary worms, their bodies carrying bristled structures suited for digging, thread themselves through the soft substrate. These animals, at their various depths, represent the base of a food web that extends from the surface of the ocean down to the floor, connected by the daily migrations of mesopelagic animals and by the slow, constant rain of particulate matter sinking from the productive surface waters above. The marine snow, as it is sometimes called, a drift of dead cells and fecal pellets and discarded mucus nets that carries organic carbon from the sunlit surface to the dark floor below. This is how the deep ocean is fed, not by the sun, which penetrates too little to reach it, but by what falls from where the sun does reach. The surface ocean, productive and warm and light-filled, subsidizes the deep ocean, cold and dark and slow, with the soft continuous drift of what was alive up there and is now falling to become food down here. And a cephalopod in the mesopelagic zone exists within this system, migrating through it, feeding from it, shaped by it. Its cold-adapted enzymes allow it to hunt efficiently at 4 degrees Celsius. Its pressure-stable membranes maintain the cell integrity that its nervous system requires. Its photophores let it communicate in the language of the zone, concealment, recognition, perhaps something like signaling between members of the same species crossing paths in the dim water. Its body is soft, its movements can be slow, its energy budget is constrained and careful. It is an animal that has met the conditions of an extreme environment through long molecular refinement, and it persists. This persistence is not dramatic. It does not involve great speed or particular strength, not in the way that predators of the shallow reef can be fast and powerful, fueled by abundant warm water food. It involves an exquisite calibration, enzyme by enzyme, lipid by lipid, photophore by photophore, to conditions that exclude almost everything else. The deep ocean is not a hostile place exactly. It is a specific one. Its hostility to the surface observer is a matter of perspective. To the animals shaped to live within it, it is simply where they are, and they are as suited to it as any reef octopus is suited to the warm, bright, food-rich waters of the surface. The mesopelagic zone receives the last of the sun's light and holds it only briefly at the top of its range. At 200 meters, on a clear day above the water, enough sunlight still penetrates to allow some photosynthesis, though less than at the surface. By 400 meters, the light is too faint for photosynthesis, though still visible to adapted eyes. By 1,000 meters, it is gone. This gradient, the slow continuous withdrawal of the sun's influence, structures life in the zone as decisively as temperature and pressure do. Animals that need light live near the top. Animals that can manage without it live further down, or migrate between the two worlds on a daily cycle, ascending to the light at night to feed on the productivity it sustains, then sinking back into safety before dawn. This daily migration is one of the largest animal movements on Earth, measured by biomass. Each evening, across every ocean, trillions of individual animals, fish, squid, shrimp, copepods, gelatinous zooplankton, rise hundreds of meters toward the surface in a coordinated ascent driven by hunger and the cover of darkness. Each dawn, they descend again. The mesopelagic zone empties and refills with them twice each day, and the carbon they carry in their bodies, metabolized from the surface waters above, is transported downward when they sink, adding to the slow flow of organic matter toward the deep. They are not doing this for the benefit of the carbon cycle. They are doing it because for each individual, rising to food at night and sinking to safety in dim daylight, has over a very long time produced more surviving descendants than any other strategy. The carbon is incidental, but it matters enormously to the chemistry of the ocean and to the ocean's capacity to absorb and store the carbon from the atmosphere above. Squid are among the most abundant of the mesopelagic migrants. Small species, many of them no longer than a finger, ascend each night in numbers that defy easy counting, filling the shallow water feeding grounds with moving bodies, then withdrawing again into depth before light comes. They are prey for surface predators, seabirds diving from above, fish rising to meet them, marine mammals descending into the upper mesopelagic in pursuit. They are also predators themselves, feeding on copepods and small fish, and on one another, and their bioluminescent photophores serve their counter-illumination function most critically during these ascents, when they pass through water still lit from above and the silhouette problem is most acute. At depth, once the light from the surface has gone entirely, counter-illumination is no longer useful. There is no background light to match. Bioluminescence at these depths functions differently, as an active lure, drawing prey toward a light in the darkness, the way a lamp draws insects to a window. Some deep-sea fish carry bioluminescent lures on modified spines extended above the head, the light dangling in the darkness in front of a mouth that waits, motionless, for whatever is drawn toward the glow. Cephalopods at depth use photophores in ways that may serve analogous functions, though the evidence here is less complete, gathered in brief observations from remotely operated vehicles descending into zones rarely visited and never easily studied. The difficulty of studying deep-sea animals in their own environment is a persistent constraint on what is known about them. Most of what has been learned about mesopelagic and deeper-dwelling cephalopods comes from dead specimens brought to the surface in trawl nets, from the stomach contents of sperm whales, and from increasingly frequent deployments of remotely operated vehicles equipped with cameras and lights. Each method has limits. Trawled animals are often damaged, their soft tissues disrupted, their coloration and posture lost. Stomach contents reveal only what an animal eats, not how it lives. And remotely operated vehicles, despite their value, descend with bright lights that illuminate the dark zone from the outside, unavoidably altering the behavior of the animals they find. The deep ocean remains one of the places on Earth where observation changes what is being observed most profoundly, and where the behavior of animals in their undisturbed state remains largely a matter of careful inference from incomplete evidence. The cold makes preservation difficult in another sense. Living specimens brought to the surface die quickly, either from the thermal shock of warming water or from the pressure reduction as they ascend. Cephalopods are among the more fragile of deep-sea animals in this respect, their nervous systems sensitive, their soft bodies subject to rapid decomposition. The few opportunities that have arisen to observe intact, recently caught deep-sea cephalopods in temporary captivity have yielded observations of lasting importance, but they are few, and the animals observed were likely not in a state that reflected their normal behavior. They were already dying, already removed from the cold and the pressure that maintained their biology, already past the conditions that shaped everything they were. This is the particular difficulty of the deep ocean as a subject. It is accessible but not comfortable, accessible to instruments and cameras, and rarely to crude submersibles, but never to the casual, patient, long-duration observation that has revealed so much about the shallow-water cephalopods studied in tanks and aquaria and on easily visited reefs. What the deep-sea cephalopod does in the ordinary hours of its ordinary day, how it rests, how it hunts without the aid of a bright spotlight announcing its position, how it moves among others of its species, what its sensory world is actually like from inside its experience. These remain questions that the available evidence frames only partially. The answers are present somewhere, at depth, in the bodies and behaviors of animals going about their lives, in cold and pressure and dim light, indifferent to the difficulty of being known. The mesopelagic zone and the waters below it are not new environments. They have existed for as long as the oceans have been deep enough to stratify into layers, and that is an old span of time. The cephalopods that inhabit them are not recent arrivals. Their lineages extend back hundreds of millions of years to ancestors that navigated ancient oceans long before the modern oceans had reached their current configuration. The molecular adaptations that maintain their membranes and their enzymes in the cold and the pressure were not designed. They accumulated, one small change at a time, in populations that persisted slightly better than populations without those changes. Across spans of time, long enough for very small advantages to become fixed traits. The deep ocean shaped them slowly, and they are the product of that shaping, as completely as any reef octopus is the product of warmth and light and abundance. What persists in the deep ocean is old. The cold has been there for a long time, and the pressure and the dark. The animals that live in it carry in their molecular structures the record of that long persistence, not as a memory, not as a narrative, but as the accumulated physical answer to the accumulated physical question the deep has been asking for as long as it has been there. This is a kind of knowing, though it is not the knowing of a mind. It is the knowing of a body written in lipid chains and protein folds and enzyme kinetics in the cold water of the mesopelagic zone at 4 degrees Celsius under 60 atmospheres of pressure, where the light from the surface is just barely gone. The firefly squid descends before dawn and holds in the dim water, its photophores dark, its body still. The pressure wraps around it, constant and unremarkable. The water is cold, and the light, what remains of it, is faint and blue, and arrives from no particular direction. And somewhere deeper still, below the last trace of filtered sunlight, in the water where nothing has ever been warm, something opens an eye, 27 centimeters wide, and holds it open in the absolute dark, patient in the way that cold and depth and long time make all things patient, waiting for the brief cold light that may be passing, even now, at the very edge of what that extraordinary eye can see. 2 fins rise from the top of the mantle. They are small and rounded, set close together near the midline, and they beat with a slow, even patience, rising and falling, in the manner of something that has never needed to hurry. Each stroke covers very little distance. Each stroke takes almost the same time as the one before it. The animal moves through the water the way a thought moves through a half-sleeping mind, without urgency, without interruption, with a quality that is not quite stillness, and not quite motion, but something that lives between the two. This is Grimpoteuthis. It drifts at a depth where no surface light has ever reached, where the water is close to freezing, and the pressure would flatten any unprotected cavity to nothing, where the seafloor below is soft sediment, and the water above is an unbroken column of cold dark. The fins are the first thing to notice about it, and for a long time they are almost the only thing, 2 pale ear-shaped structures, moving in a rhythm so regular it seems almost mechanical, lifting and pressing, the whole body rising and settling in a gentle vertical oscillation that carries it forward by the smallest of degrees. The webbed arms hang below. They spread in a loose arrangement, the membrane between them giving the animal a wide, draped quality, like a soft bell, or like the hem of something worn loosely and allowed to trail. The skin over the mantle is pale and smooth, and faintly translucent at the edges, and in the beam of a research submersible's light, it takes on a color that is difficult to name, not quite white, not quite gray, something cooler and more even than either. The body is soft throughout. There is no hard shell, no rigid structure, no armored surface. Everything about this animal yields to the water around it. The genus Grimpoteuthis belongs to the family Opistoteuthidea, a group sometimes called the umbrella octopuses, for the wide, sail-like web that connects their arms from base to tip. Within that group, approximately 16 described species carry the name Grimpoteuthis*, each one distinguishable from the others by the small particulars of fin shape, arm proportion, and the texture of the skin. What they share is the paired fins, the soft body, the deep range, and the quality of motion that has given them their common name, the Dumbo octopus, for those fins rising and falling like the ears of a large, patient fictional animal. The name has followed them since the first specimens were described, and it is not an unkind one. There is something in it that fits. These animals differ from their shallow water relatives in a structural detail that matters enormously to how they move through the world. Most octopuses propel themselves primarily through jet propulsion, drawing water into the mantle cavity and expelling it sharply through the siphon, a mechanism that is fast and effective and suited to the quick reactive life of a reef or a rocky shore. Asterisk Grimpoteuthis asterisk retains the siphon and can use it, but the fins have become the primary engine. In the deep and quiet water where these animals live, where currents are slow and prey does not require pursuit, the slow beat rhythm of the fins is enough. It is more than enough. It is precisely the right way to move in a place where efficiency matters more than speed, where the cost of every calorie must be weighed against the difficulty of finding the next one, where the water offers no quick escape to something that moves quickly, and every advantage to something that moves well. The arms, though not the engine, do their own work. Running along each arm, on the oral surface, the side that faces inward toward the mouth, are fine, hair-like structures called cirri. Each one is small, a few millimeters at most, and they line the arms in dense rows between and alongside the suckers. They are sensory structures, wired to detect chemical and mechanical information from whatever the arm is touching or passing near. When a Feltus asterisk Grimpoteuthis asterisk moves close to the seafloor, when the tips of its arms trail across the soft sediment, the cirri are reading that surface continuously, the texture of it, the chemical signature of it, the faint disturbances in the water that might indicate something living nearby and small. The arms do not need to grip and manipulate to gather information. They gather it just by being present, by moving slowly through a medium that is carrying at all times the faint traces of everything that has recently passed through it. This is one of the features that separates asterisk Grimpoteuthis asterisk from the incerate octopuses, those species without cirri, which include the shallow water species better known from reefs and rocky coastal habitats. The serrate octopuses, the group to which asterisk Grimpoteuthis asterisk belongs, have carried these sensory filaments with them into the deepest habitats available to any octopus on Earth. At 4,000 meters, at 5,000 meters, the cirri are still there, still reading, still useful, a sensory architecture that has traveled down with these animals across the long history of their adaptation to pressure and cold and dark. The prey that the cirri help locate is modest in scale. Asterisk Grimpoteuthis asterisk feeds on polycate worms, those segmented, bristled animals that thread through the sediment of the deep sea floor in numbers that are larger than they might seem from above. It feeds on amphipods, small crustaceans that are among the most abundant animals in the deep sea, found at every depth and in nearly every habitat. It takes copods, isopods, and occasionally small bivalves when they are available. None of these require the strong, decisive beak strike that a reef octopus uses against a crab. Instead, asterisk Grimpoteuthis. Asterisk is thought to swallow small prey whole, or nearly so, a feeding strategy consistent with the softness of its body, the modest size of its beak, and the generally small scale of what it eats. The meal requires no violence. It requires only proximity, a surus tip reading the right chemical gradient from the water, an arm bending gently downward towards something that has no warning of what is coming. The deep sea floor where asterisk Grimpoteuthis asterisk hunts is not featureless, though from a distance it might appear so. There are small variations in the sediment, patches of coarser material where a current once moved across the plain, areas where different organisms have churned the top layer. The occasional piece of hard substrate, a fragment of shell, a bone, a piece of volcanic rock that settled from the ridge far away. It is a sparse landscape measured in the scale of what it offers to a small animal, but it is not empty, and through it, at the pace of those steady fin beats, *Grimpeteuthus* moves, not covering much ground in any given hour, not needing to. The arms hang down, the surrey reed, the fins rise and fall. The water at this depth holds the cold with a permanence that nothing seasonal can disturb. At 3,000 meters, the temperature sits close to 2 degrees Celsius, sometimes a little below, sometimes a fraction above, varying by almost nothing across years and decades. It is a cold that is not like the cold of a winter morning or the cold of a plunge into a mountain lake. It is a cold that is absolute in the sense that it has no counterpart, no warmer season waiting, no shallower layer, a quick swim away. Everything that lives here has been built for this temperature, tuned to it at the molecular level, dependent on it in ways that would become liabilities anywhere warmer. The enzymes inside a *Grimpeteuthus* cell function well at 2 degrees Celsius. They do what they need to do. They process what they need to process with a steady competence that would not translate to warmer water. These are cold-adapted proteins, shaped by a very long time in very cold water into tools that work precisely in the conditions where they are used. The pressure at these depths is something the body of *Grimpeteuthus* carries without apparent effort, because it has no choice, and never had. An animal raised in shallow water and lowered to 3,000 meters would not survive the change. The gas-filled spaces in a vertebrate body would be compressed past the point of function. The cell membranes would stiffen. The enzymatic chemistry would fail. But *Grimpeteuthus* has no gas-filled spaces to compress. The body is soft and fluid throughout, the same pressure inside as outside. The cells built with the molecular architecture to function under loads that would destroy a surface animal completely. The pressure is not a challenge, it is overcoming. It is simply the weight of the world this animal has always known. Very few observations of *Grimpeteuthus* in its natural environment exist. These animals live at depths that require hours of transit time in even the most capable research submarines, and the dives themselves are costly and rare. What footage does exist, gathered over decades by remotely operated vehicles and manned submersibles deployed from oceanographic research vessels, tends to show the same quality of behavior, the steady fin beat, the slow forward progress, the arms trailing below, the occasional approach toward the bottom to investigate what the surrey have detected. The animals do not flee from the lights of approaching vehicles with any great urgency. They hold their position. They continue their fin's slow rhythm. They appear to regard the vast glowing machine descending toward them with the same measured patience they bring to everything else. This composure in the face of something entirely outside their evolutionary experience is not courage, and it is not indifference. It is likely a reflection of the deep-sea reality that fast reactive behavior is expensive in a place where metabolic resources are scarce, and that in an environment without quick visual predators, where the threats that exist are mostly chemical and mechanical and slow, there is simply no advantage to a hair-trigger startle response. The calm of *Grimpeteuthus, in the submersible's light, is the calm of an animal whose whole history has been written in slow water, where the adaptive value of stillness and patience is higher than almost anything else. One fin stroke, then another, then a long pause in which the body drifts slightly upward before the next stroke corrects the drift. The pattern is almost hypnotic in its regularity. Watching footage of a Pteris grimpeteuthus asterisk moving through open water, it is difficult to track the passage of time. The motion is so even, the environment so uniform, the animal so unchanged from one moment to the next, that the mind releases its usual grip on sequence and simply settles into the rhythm of what it is watching. The reproduction of asterisk grimpeteuthus asterisk has taken longer to understand than many aspects of its biology, partly because opportunities to observe it directly are so limited, and partly because what has been found does not follow the pattern expected from shallow water octopuses. In most octopus species, a female lays a clutch of eggs, tends them through a single synchronized developmental period, and dies shortly after the eggs hatch, one reproductive event, concentrated and complete. What appears to be happening in asterisk grimpeteuthus asterisk is something different and considerably more difficult to interpret. Females carry eggs in multiple simultaneous stages of development. One female may have eggs that appear newly deposited alongside eggs that are nearly ready to hatch at several intermediate stages between them, all present at once, all being tended at the same time. The significance of this arrangement becomes clearer when considered against the deep sea environment. Food in the deep ocean does not arrive on a seasonal schedule. The rain of organic material descending from above is intermittent and unpredictable. The density of prey organisms varies without reliable pattern, and the metabolic cost of reproduction is high relative to available resources. A single synchronized reproductive event, timed to coincide with a bloom of prey or a seasonal increase in food availability, makes sense in a shallow coastal habitat where such seasons exist. In an environment without seasons, the strategy that fits is a continuous one, producing eggs gradually, maintaining them in multiple developmental stages, means that at any given time, some young are close to ready, and the energy invested in reproduction is distributed across time rather than concentrated in a single costly event. This interpretation was described in detail in a 2021 paper by Henk-Jan Hoving and colleagues who examined the reproductive biology of asterisk grimpeteuthus, asterisk individuals collected over years of deep-sea sampling. The picture they assembled was of females that appear to produce eggs on an ongoing basis, attaching them to hard substrate where available, a piece of coral skeleton, a fragment of rock, any surface solid enough to anchor them against the slow currents of the deep. The eggs are large relative to the animal's body, encased in a tough protective layer, and they take a very long time to develop. How long exactly is not known with precision, because monitoring a specific set of eggs at 4,000 meters across months or years is not yet within the practical reach of oceanographic research, but the developmental time is thought to be extended, consistent with the cold temperature slowing every biological process, and the large, well-provisioned eggs suggesting the investment of substantial maternal resources. Hard substrate is not abundant on the soft sediment plains where many asterisk grimpeteuthus asterisk individuals are found. When a suitable surface is located, a piece of rubble, a dead coral branch, the shell of a large bivalve, it may hold eggs from multiple depositions at different stages. A pale cluster of newly laid eggs, smooth and evenly colored, may sit close to a set of larger eggs that have darkened somewhat, the embryos inside them grown enough to be visible as a denser mass within the case, and beside those a third group that are darker still, more developed, the internal shape of a small animal pressing faintly against the outer membrane, all of them tended by a single female, all of them at different points along the slow arc from cell to animal. The patience required is not the patience of a waiting mind. It is the patience of a body that has been tuned, over a very long time, to the particular tempo of the deep ocean. The hatchlings, when they emerge, are not helpless. This was documented clearly in 2021 by Tim Shank and colleagues at the Woods Hole Oceanographic Institution, who were fortunate enough to observe a newly hatched Grimpateuthis individual in sufficient detail to assess its developmental state. What they found, and what their footage and measurements confirmed, was an animal that emerged from the egg case already in an advanced state of readiness. The fins were present and functional. The arms were developed, with cirri already in place along their oral surfaces. The eyes were open. The mantle was formed. Within moments of leaving the egg, the hatchling was swimming, the same slow, even fin beat of the adult, in miniature already operating. There is no larval stage in the conventional sense. There is no period of helplessness during which the young animal depends on some reservoir of nutrition it cannot yet supplement. No drift phase, during which it is carried by currents it cannot control. No slow and vulnerable assembly of functional systems while the world waits to take it. The egg has done all of that work already, inside its tough protective case, over the long, slow months of development. The hatchling arrives as a small, complete, functioning version of what it will be throughout its life. The fins beat, the arms trail, the cirri read whatever is in the water around it. It is, from the first moment, exactly what it is. This precocial emergence, the term for hatching or birth into a fully capable state, is not unusual in cephalopods as a group. Many octopus and squid species produce young that are immediately functional, but in Grimpateuthis*, the significance of it is amplified by the environment. In the deep ocean at 4,000 meters, there is no shelter for a helpless animal, no nursery habitat, no seasonal bloom of food, time to support a vulnerable life stage. The investment of resources into large, well-provisioned eggs that develop slowly and completely before hatching is a strategy that matches exactly the conditions in which the hatchling will have to live. It emerges ready because the deep ocean offers no accommodation to anything that is not. The hatchlings seen in the 2021 footage moved away from the substrate within moments. The fins were already beating in the same rhythm, rising and falling, that they would maintain across whatever life lay ahead. There was no hesitation visible, no period of orientation, no slow realization of what the water around it was. The animal simply moved into it, as though it had always known this place, which in some sense it had, all of its development occurring at depth, its entire formation shaped by cold and pressure, its first breath of open water taken in the same environment where the egg that made it had sat for months on a piece of hard substrate, in the dark. The ROV that recorded it was almost certainly the first light that young animal had ever encountered. The camera approached from above, drawn in by the small motion of those newly active fins, and held its position a short distance away. The hatchling did not flee. It did not alter its course in any way that was clearly reactive. It continued moving through the water at the same slow, deliberate pace, the fins beating, the arms hanging below, already the same kind of patient, unhurried presence that every Grimpateuthis* individual appears to be, at every depth and age and size where they have been observed. The research vessel above them on the surface would have been in daylight or night depending on the hour, its deck lit and its instruments running, the cable connecting it to the ROV hundreds of meters long and pulling gently with the movement of the ship in the swell. None of that reached the hatchling. The surface is a different world entirely, separated by a water column so long and cold that it may as well be another planet. The hatchling was already home. It is worth pausing here on what the fins achieve that jet propulsion cannot. At the metabolic rates required for sustained jet propulsion, drawing water in, contracting the mantle forcefully, expelling it, the energy cost is significant. For a shallow water octopus that eats frequently and lives in a rich environment, this cost is manageable. For an animal living at depths where a single meal may not be followed by another for days, where the body temperature never rises to accelerate digestion or replenish energy stores quickly, where every calorie must serve as long as it can, the fins offer something essential, a way to move that does not spend much. The slow beat of those paired structures generates lift and forward motion at a cost that is a fraction of what a mantle contraction requires. The animal can patrol the seafloor, move between feeding sites, carry eggs to suitable substrate, investigate what the cirri detect, all of this powered by a motion so economical it barely registers as expenditure. The jet remains available for the rare moments when speed is needed. But those moments are rare, and the fins are always moving. There is a quality to the observation of *Grimpatoothus, asterisk footage, that is different from watching the footage of most other animals. With a fish, there is the constant motion of fins and tail, the quick corrections of direction, the reactive darts and pauses that keep attention alert. With a reef octopus, there is the continuous color change, the arm exploration, the shifting and testing of the immediate environment. With *Grimpatoothus*, there is the fin beat, and then there is the fin beat again, and the arms trailing, and the pale body in the dark water, and nothing else. The consistency of it produces a kind of visual calm that mirrors something internal, as though the animal's outward presentation and its metabolic reality are perfectly aligned, that what it looks like from outside is exactly what is happening inside, which is a slow and steady and unhurried process of being alive in very cold water. The arms move sometimes. One will extend downward when the cirri have detected something worth investigating, lengthening gradually until the tip touches the sediment, and then it will move across the substrate in a slow sweep, the cirri reading the surface, the arm remaining extended for a moment before withdrawing back into the draping arrangement below the mantle. It is not the arm behavior of a shallow water octopus solving a problem, the focused, exploratory, multiply-corrected manipulation of something being assessed for its properties and potential. It is more passive than that. It is the arm responding to what the cirri have already found, following the chemical gradient toward its source, and then either closing on something or not, and then returning to rest. What *Grimpatoothus* does not do, so far as any observation has revealed, is the suite of behaviors that mark the shallow water octopus as an unusually active cognitive presence: the tool use, the jar opening, the navigation of novel environments, the play behavior, the individual personality differences that make one animal recognizably different from another in captivity. Whether these abilities are simply unobserved, or are present in modified form, or are genuinely absent, whether the cognitive architecture that supports them is different in a cirri deep-sea octopus than in its in-cirried shallow water relatives, is a question that remains open. The observations available are too few, too brief, and too constrained by the difficulty of working at depth to allow any firm conclusion. What is known is behavior, and what the behavior shows is an animal that is exquisitely fitted to its world, moving through it with a patient efficiency that leaves nothing wasted. The range of *Grimpatoothus* asterisk extends across ocean basins. These animals have been found in the Atlantic and Pacific and in other deep ocean regions, always below 1,000 meters and often much deeper, some records extending to nearly 5,000 meters. The 16 or so described species occupy different parts of this range, some more reliably encountered in certain ocean regions or at certain depth intervals. The full distribution is probably wider than the records suggest, because the records depend on where research submersibles have gone, and where research submersibles have gone is a small fraction of where the deep ocean exists. There is a very large amount of cold, dark ocean at 3 to 5,000 meters depth. It covers more of the planet's surface than all the land masses combined, and in most of it, no human instrument has ever been deployed. Asterisk Grimpeteuthus. Asterisk may be present across all of this in densities that cannot be guessed from the handful of individuals observed in well-sampled locations. Or it may be patchier than that, concentrated around particular substrate types or food resources that are themselves distributed unevenly. No one knows. The footage and the specimens provide a picture of what these animals are, but not of how many there are, or where exactly they all go, or how their populations are organized across the immense dark volume of the deep ocean that is their home. What the footage does provide, returned from those expensive and infrequent dives, is the image itself, the fins moving, the body pale in the light, the arms trailing below. Sometimes the animal is close to the bottom, and sometimes it is in mid-water, drifting at a height of several meters above the sediment. Sometimes it is moving with apparent direction, and sometimes it appears to be simply maintaining position, the fins providing just enough force to hold steady against the faint drift of the current. The face, if it can be called that, the anterior surface of the mantle around the mouth and eyes, is mild and uniform. The eyes themselves are moderately sized, not the enormous structures of some deep-sea cephalopods that have invested enormously in visual acuity. The world of Asterisk Grimpeteuthus. Asterisk is sensed largely through the arms and their cirri, through chemical and mechanical information gathered in close contact with the substrate. The eyes may play a secondary role, useful for detecting the bioluminescent flashes of nearby organisms, but the primary sensory work is done lower down in the slowly moving tips of the arms. The egg cases on the hard substrate, when they are found, tend to be found in clusters, or near the same piece of rubble across multiple samplings, which suggests that females may return to productive attachment sites, or that the same sites are favored by multiple individuals across time. Whether this represents any form of site fidelity, any memory of useful locations, is not established. The deep-sea environment may simply constrain where hard substrate is available enough that the same limited sites are used repeatedly by whoever finds them. The distinction between learned preference and constrained choice is difficult to make from the outside, and especially difficult to make in an environment where observation is so limited. What is established clearly and in detail is that the eggs themselves are carefully placed. The attachment is secure. The cases are anchored to the substrate in a way that resists the slow movement of bottom currents without detaching them from the animal that tends them. A female carrying eggs at multiple stages is not simply releasing them into the water to drift. She is maintaining them attached on a specific piece of substrate in a specific location for whatever extended period it takes for the slowest of them to develop to the point of hatching. The investment is real, and it is long. The hatchling that departs is the end product of all of it. The months of slow development in the cold, the sustained attachment to hard substrate, the gradual investment of yolk reserves into the forming animal inside the case, the patient maintenance of a set of eggs at different stages, some nearly ready, some just beginning. All of it culminates in a small animal with beating fins and trailing arms, and Siri already reading the water, already moving away from the place it was made, into the same cold dark water where every asterisk Rimpetoothus asterisk has always lived. The fins rise, they fall, they rise again. The animal moves forward by the smallest of degrees, in water that is almost freezing, under a pressure that would be incomprehensible to anything not built for it, at a depth where the only light that has ever arrived has come from the bodies of other organisms, or in the last century and a half, from the instruments of a species that has barely begun to look. The Surrey trailing from the arm tips read the water as the animal passes through it. There is a faint chemical trace here, something biological, something that was living recently, or is living still, somewhere in the direction the current is coming from. The arm tips curve slightly toward it. The fins continue their rhythm. The body drifts forward. The pale mantle catches the last edge of the submersible's retreating light, holds it for a moment as a faint luminescence at the margins, not quite visible, not quite gone, and then the darkness takes it back, completely and without drama, the way the deep ocean has always reclaimed what passes through it. The fins move in the dark, they continue. They have always continued, in water this cold and this old, in a silence that has no floor, long after the last instrument has gone, and the surface world has returned to its ordinary distance above. The skin lies still against the rock. It is pale, almost colorless, flattened smooth over the surface of the stone, as though it has always been there, as though it grew from the same material as the seafloor and belongs to it. Nothing moves. The water above turns slowly in a current too faint to see, and the light comes down from above in long, slow columns, shifting without pattern, and the skin beneath it shows nothing. Then a single papilla rises. It is small. It takes less than a second. At the center of the dorsal surface, one small column of skin pushes upward from the flat, a fraction of an inch at most, blunt-tipped, slightly darker than the tissue around it. It stands there, perfectly still. Then another rises a little to its left, slightly taller, and another beside that one, and the skin that was smooth begins very slowly to roughen, not all at once but in a quiet progression, one small structure after another finding its height, the surface accumulating texture, the way a dry landscape accumulates shadow as the sun moves, gradually, and then all at once the whole character of the skin changed. The animal it belongs to has not moved. These structures are called papillae. Each one is a column of skin shaped by radial and circular muscles working in opposition, one set drawing the tissue outward, the other controlling the width and angle of what rises, and they work fast, faster than most muscle systems in the body can coordinate, because what they are doing requires matching a surface that is already there, already detailed, already complex in 3 dimensions, and the match must be close enough to make the whole animal disappear. From a distance of a foot or two, looking at the right angle, an octopus deploying its papillae in full becomes part of the substrate beneath it. The same body that a moment ago lay soft and pale is now a piece of reef. The transition happens in under a second. The octopus has not gone anywhere. It has simply become, for as long as it chooses, something else. What the skin does in that second belongs to a different order of speed than what the brain does. The pattern for the match, the specific configuration of papillae that will replicate this particular rock's particular texture, does not have to travel far. The signal goes out from the central nervous system, but the execution is local. the fine adjustments handled by the motor neurons of the arms and the mantle surface itself, close to the muscles they control, so that by the time any conformation has returned to the brain, the transformation is already complete. The skin does not wait for approval. It acts from its own kind of knowing, distributed and immediate, a knowing that lives in the tissue. Across the body surface, millions of small cells sit ready. The chromatophores are the ones most often described. Sacs of pigment, brown, red, yellow, black, each one attached to its own small fan of muscle fibers, each one wired to a motor neuron, so that when the signal arrives, the sac expands from a pinpoint to a disc in a fraction of a second, and the color it holds floods outward across the skin. These are the fast performers, the cells that make the flicker and the pulse and the rapid whole-body color shift look almost instantaneous when viewed at normal speed. Beneath them, embedded a layer deeper in the dermal tissue, are other cells with a different mechanism and a slower register. These are the iridophores, and what they do with light is not simple pigment release, but interference. An iridophore holds within it a stack of thin reflective plates, each plate a fraction of a wavelength of light, thick, separated by spaces of a precise and adjustable width. Light entering the stack bounces between the plates, and the waves that return from different layers either reinforce each other or cancel, depending on the spacing, and what emerges from that interference is structural color, not a pigment color absorbed and re-emitted, but a color produced by the geometry of reflection. The iridophores in the skin of certain squid have been documented reflecting wavelengths that extend beyond what the human eye can receive, into the near ultraviolet. The skin, from the perspective of an animal that can perceive those wavelengths, carries information invisible to almost every other creature watching from the outside. The tuning of the iridophore plates is not fixed. Neurotransmitter molecules diffusing through the tissue alter the spacing of the reflective layers, shifting the interference pattern, and with it the color reflected. The change is slow relative to the chromatophore's snap. It moves across seconds rather than fractions of a second, and it gives the skin a secondary layer of color change that operates beneath the fast surface display, a deeper and quieter modulation happening in parallel with the rapid surface performance. When both systems are operating together, the skin of the animal is doing at least two things at once, one fast, one slow, one visible to most observers, one not, and there may be a third thing. In the skin of the octopus, embedded in tissue far from the eyes, opsin proteins have been found. Opsins are the molecular foundation of photoreception, the proteins that detect light by changing shape when a photon strikes them, triggering the chain of events that eventually produces a visual signal. In the eye, they are organized into tight arrays of receptor cells connected to the optic nerve, and everything they detect feeds into the brain's picture of the world. In the skin, they sit in tissue that has no direct connection to the optic lobe, no obvious channel through which what they detect could become an image. What they are doing there is not fully understood. The honest answer is that no one knows yet, and the work to find out is ongoing and unresolved. One possibility, and it is a possibility only, is that the skin is in some diffuse and partial way responsive to light across its whole surface, not seeing, not forming images, but detecting something, intensity or direction, or some quality of the light falling on it that helps calibrate the color it produces. The octopus is famously colorblind. Its eyes, for all their structural sophistication, contain only a single type of photoreceptor, which means they cannot distinguish wavelengths, the way a trichromatic eye does, cannot in principle see color. And yet the camouflage they produce is color matched, often closely, to the substrate around them. The skin opsons are one part of the attempt to understand how this can be true. The skin is not the eye, but the skin is not entirely in the dark either. Let the image of the opsin settle, that small and ancient protein, the same molecule that underlies sight in insects and fish and the waking human eye, present here in a patch of tissue that has never been thought of as a visual organ, doing something not yet named. There is something in this that resists the clean categories. The octopus's body does not divide itself along the usual lines between sensing and moving, between surface and interior, between what perceives and what acts. The skin moves in response to what the eyes see, but it may also sense in its own right. It is a boundary that is not entirely a boundary, a rock face at 5 feet below the surface. The light comes in from above at a slight angle, striking the kelp-covered stone and catching on the edges of encrusting organisms, the pale rims of tunicates, the hair-thin stalks of hydroids, the irregular bumps of sponge and coralline algae. The octopus occupying a hollow in the rock's surface is not visible. What is visible is the rock, or rather a section of rock slightly different in texture from what surrounds it, though a careful eye held close enough might eventually notice the 2 dark pupils resting in the surface, watchful and level, giving nothing away. The skin displaying this pattern received instructions and carried them out in under 200 milliseconds. The pattern is not unique across the range of all the environments an octopus encounters. The configurations its skin takes on group into a smaller number of named templates, 3 foundational types described and documented by researchers who spent long years photographing cephalopod camouflage under controlled and natural conditions. There is the uniform pattern, an even, unpatterned tone across the body, offering general background matching when no specific texture is needed. There is the model, a broken, patchy arrangement of lighter and darker regions that interrupts the body's outline without fully disrupting it. And there is the disruptive pattern, high contrast, large-scale irregular patches of dark and light that break the body's shape into pieces no eye can easily reassemble into an animal. Each pattern serves a different visual environment. Uniform works against backgrounds that are themselves uniform: open water, bare sand, flat pale rock. Model works against the mid-complexity of reef surfaces and mixed substrates where some texture variation helps, but no strong contrast is needed. Disruptive works against high contrast, complex visual scenes where the animal's outline would otherwise stand out. Dense algae, broken rubble, the chaotic overlap of reef organisms at close range. The octopus does not deliberate between them. The nervous system reads the visual environment, compares it against something, and selects the appropriate template in a time shorter than a blink, and then the transition begins. High-speed video has captured the pattern change in cuttlefish, closely related to the octopus, with the same chromatophore system and many of the same behavioral capacities, and what the video shows is not what intuition predicts. The change does not happen all at once, the whole body shifting from one state to another simultaneously. It propagates, it moves. A wave of change travels across the body surface, beginning at one edge and rolling to the other. The leading edge of the new pattern advancing steadily while the old pattern holds behind it, the 2 states coexisting for a fraction of a second across the midline of the body, before the new one reaches the far edge and the transition is complete. The whole body rewritten in under 1/5 of a second, and the rewriting visible as movement, a slow lateral sweep of color and texture crossing the skin like the shadow of a cloud moving over a hillside. This is not what a simple on-off switch produces. The traveling wave requires that different regions of the body receive their signal at slightly different times, or process the same signal at slightly different speeds, or are coupled to one another in some way that causes the cascade to spread like a front rather than a single simultaneous event. It means the skin change is not just a color change, but a coordinated dynamic event, the body participating in its own concealment as an integrated and moving system, not as a collection of cells, each responding independently to the same broadcast command. The wave crosses the skin, and the animal is gone. Not gone. There, still, perfectly still in its hollow in the rock, the skin fully settled into its disruptive pattern, the papillae holding their heights, the chromatophores locked open or closed in the configuration that makes this animal invisible to the eyes of the animals that hunt it. The octopus waits. Time passes in the way it passes in water, without markers, without the punctuation of sound or light change, the current moving very slowly over the still skin of an animal that has erased itself from the visual world. The eye that looked down on this scene from outside the water would find nothing. It is a strange kind of presence, to be there, to be perceiving, to be processing the sensory input of the arms resting against the rock and the light falling on the skin and the chemicals dissolved in the water drifting past the suckers, and to be, from any outside perspective, simply absent. The skin that achieves this is not just a surface. It is in a sense the primary interface between this animal and the world it lives in, the place where camouflage and communication and sensation all converge, where what the octopus is to the world outside and what the world outside is to the octopus are both written continuously and simultaneously. There are other conversations the skin conducts that have nothing to do with concealment. When two octopuses meet, an event rare enough in most species that each encounter carries some weight, the skin produces patterns that are not found in the camouflage repertoire. The pulses and waves and traveling displays that move across the body in these moments are not background matching. They are addressed. They are sent. What they contain is debated, and the precise semantics of cephalopod skin displays in social contexts is an area where the science is still building its vocabulary. What is clear is that the patterns produced are different in character from camouflage, produced in different contexts, and that they influence the behavior of the animal that receives them. The polarization channel may carry some of what these displays contain. The octopus eye contains photoreceptors arranged in two orthogonal orientations at 90 degrees to each other, so that the eye is sensitive not only to the intensity and wavelength of light, but to the plane in which it oscillates. This is polarization sensitivity, and it gives the octopus access to a visual channel that most other animals cannot read. The iridescent skin of the octopus and its cephalopod relatives strongly modulates the polarization of the light it reflects, so that two animals meeting in the water are exchanging information in a dimension invisible to the fish watching from 10 feet away. What the messages say is not fully known, but the channel is real, the mechanism is established, and the skin is its medium. The rock wall at 2 feet below the surface holds an octopus that has been still for several minutes. The arms rest loosely across the stone, their suckers in light contact with the surface, the chemoreceptors in each sucker sampling the chemistry of whatever they touch. The skin is fully patterned. The eyes move, slowly, scanning the water ahead. A small crab crosses the open sandy floor 4 feet below, pausing, moving again, entirely unaware of the animal watching from above. The octopus gathers itself, a subtle change in the posture of the mantle, a shift in the arrangement of the arms, nothing visible to anything watching, and then it moves. The skin changes as it moves. Not before, during. The pattern that served concealment against the rock is already beginning to shift before the animal has fully left its hollow, the chromatophores adjusting, the papillae slowly lowering, the skin reading the changing visual context in real time and producing the configuration appropriate to what the eye is seeing now rather than what it was seeing a moment ago. The camouflage is not a costume put on and taken off. It is a continuous response to the world, a living display generated freshly from moment to moment, as long as there is light for the skin to read and eyes to compose an answer to. This capacity to read a surface and replicate it, to read a context and shift the display, to produce a traveling wave of pattern change in under 200 milliseconds, runs through the same tissue that may also sense light directly, that exchanges polarized signals with other individuals, that processes tactile and chemical information through the suckers and the cirri, that in certain species produces structural colors reaching into wavelengths no vertebrate can see. What the skin is doing at any given moment is the sum of all of these things at once, and none of them stops for the others. Deep in the tissue, one layer below the chromatophores, an iridophore adjusts its plates. The spacing shifts by a fraction of a wavelength. The interference pattern changes. What was reflecting in the visible green shifts toward a color shorter and colder, a blue edging toward wavelengths that leave the range of human perception. Nothing changes in the scene as a person looking down into the water would read it. The octopus's coloring looks the same, but in the water around it, a signal is being broadcast that the octopus's own eye, with its polarization-sensitive, wide-spectrum photoreceptors, may be reading in a way no human instrument positioned outside the scene would think to measure. The crab stops moving. The octopus hangs motionless above it, a foot of water between them. The skin is still. Then the pattern changes, fast, the disruptive patches snapping out in the high-contrast configuration of a hunting animal that no longer needs to be invisible. The decision made somewhere in the distributed brain, carried out across the full body surface in the same fraction of a second it always takes, and the octopus drops. The encounter is brief. The crab is held, and the octopus moves back toward the rocks with the food carried loosely in the folded web of its arms, the skin already returning to its mottled, textured concealment pattern as it goes, the traveling wave moving from head to tail as the animal settles once more against the stone. By the time it is still again, the skin has already remade itself. What remains on the seafloor is a piece of sand and a patch of water, and no sign at all that anything was there. The skin has closed over the event. There is something very particular about what the surface of this animal does that is easy to miss if the attention stays on the behavior and does not move down into the tissue. Most animals have skin that is inert between behaviors, a covering, a barrier, a thing that holds the animal in the world out. The skin of the octopus is never quite inert. Even at rest, in full concealment, the chromatophores hold their state with active muscle contraction. The color of the skin is not a passive thing, but a continuously maintained posture, requiring energy, requiring neural input, requiring the distributed nervous system to keep saying, hold this, hold this, hold this. When an octopus dies, the skin patterns collapse almost immediately, the chromatophores retracting to their smallest state, and the body fades to pale. The color was not a property of the tissue, it was a behavior of the tissue, ongoing, chosen, sustained. The skin is doing something, always. In the water at night with the light gone, an octopus moving across the reef is producing pattern changes that no eye outside its own is likely to be reading. The camouflage in the dark is unnecessary. And yet the chromatophores continue. The patterns change. The waves travel. Whether this is residual activity or the discharge of a system that runs continuously by its own logic, or some form of display addressed to a darkness that nothing is watching through, is not known. It has been observed. It has been filmed. The octopus in the dark makes patterns on its skin, and no one knows for whom. The brain sends the signal, and the signal does not stop. Along the vertical lobe of the supraesophageal brain, that small, dense structure that sits near the center of the animal's neural mass, the part most specifically associated with the consolidation of what the octopus has learned and encountered and done, the day's events move through at some cellular level, not yet fully legible to the instruments that watch it. Something about what the skin saw and matched and produced. Something about the crab and the drop and the return to the rock. Whether this constitutes experience in any form, the word experience usually implies, is a question that lives far outside what the current science can answer, and the honest position holds it open without forcing a resolution. What is known is this: the events of this day will alter the animal's future behavior, the precision with which it matches a substrate, the speed with which it selects a pattern template, the angle from which it approaches prey. All of these are learned, all of these are refined across a life, and all of them depend on the consolidation of past events into something the animal carries forward. That consolidation requires the vertical lobe, a structure no larger than a peppercorn, sitting at the center of a distributed mind, turning what the skin and the arms and the eyes have encountered into something that will still be available tomorrow and the day after and across the weeks of a short life. The rock face is still, the skin is still, the papillae are lowered, the chromatophores hold their modeled configuration without effort, or rather, with the continuous quiet effort of maintained contraction, the effort so ordinary and so constant that it recedes entirely into background, indistinguishable from stillness. The octopus rests inside its concealment, which is also its presence, which is also its body. The water moves slowly above. All the small systems that make the skin what it is continue working in the dark behind the visible surface. The opsin proteins in the dermal tissue sit in their positions, available. The iridophore plates in their stacked arrays hold their current geometry. The chromatophore muscles hold their current state of contraction or relaxation. The papillae lie flat against the mantle, their radial and circular muscles at rest, waiting. The suckers along the underside of the arms continue their slow chemical sampling of the rock. The chemoreceptors there, sending small signals into the axial nerve cord of each arm, which processes them locally and does not always forward them upward to the central brain. The arms know the rock knows the arms. The exchange continues without announcement. The water above is darker now, the light coming from above thinner and more oblique, the day moving toward its end. On the sandy floor nearby, the pale trails left by the crab's passage are already being smoothed by the slow work of the current, the sand grains settling and resettling, the surface returning to its undifferentiated state. Whatever happened here is leaving no long record. The light thins further. In the tissue of the skin, the opsins wait. In the iridophore layers, the plates hold their geometry, ready to be shifted by a molecule of neurotransmitter diffusing through the fluid between cells, ready to change the interference and change the color and change what signal the skin is carrying through the polarized channel toward whatever is close enough to receive it. The chromatophores hold, the papillae lie flat, and the whole surface, the whole body surface, from the dorsal mantle to the tips of the arms to the webbing between them, continues its continuous negotiation with the world outside it, a negotiation conducted in light and texture and chemistry, in frequencies some eyes can read and others cannot, in patterns that persist through darkness, because the muscle that holds them does not need light to hold. The water darkens, the current slows, somewhere in the distributed mind of the animal, in the arms resting against the rock, in the optic lobes holding the last images, the eyes gathered before the light became too thin to work with, in the vertical lobe settling the day's events into whatever form they will take when they are encountered again. The pace of processing adjusts to the dark and the quiet. The skin maintains its pattern as it always does. The pattern is the same pale, mottled brown it has been for the last hour. The papillae long since deployed and forgotten, the color continuous, the surface still. From above, from outside the water, there is nothing to see but rock. The distinction between the rock and the animal resting against it has been erased so thoroughly that even the question of where one ends and the other begins becomes, in this light and at this distance, genuinely uncertain. The octopus is inside its skin. The skin is inside its pattern. The pattern is inside the rock face, and the rock face is inside the ocean, as it has been for longer than there have been animals to rest against it, as it will continue to be long after this particular animal and this particular evening are gone. The ocean holds all of it with the same indifferent and steady patience. The light finishes fading. What was dim becomes dark. What was dark becomes the particular darkness of shallow ocean water at the end of a clear day, not the absolute darkness of the deep, but a rich and textured black, shot through with the faint phosphorescent blue of bioluminescent plankton, where the water stirs, lit briefly and randomly, the light appearing and disappearing without pattern or purpose. The octopus in the rock does not move. The skin holds its pattern in the dark, as it always does, the chromatophores sustaining their positions through some quiet continuity of neural signal that runs on beneath whatever else the brain is doing, beneath whatever passes through the vertical lobe in these still hours. The pattern the skin holds in the dark is addressed to no eye. The rock it rests against does not see. The water moving over it does not see. The small organisms growing on the stone beside the octopus's arm do not see. The pattern is held because holding it is what the skin does, because the signal that says hold this, hold this, hold this has not stopped and will not stop until the animal chooses something else, or until the animal is gone. It is an expression without an audience, a maintained statement in a room where no one is listening, and it is perhaps the truest image of what the skin of this animal is. Not a display, not a camouflage, not a communication system, but all three at once, continuously, because the distinction between those things has never been as clean in this animal as it seems from the outside. The skin is not for anything. The skin is what the animal is. The water moves over the rock in slow, even breathing, or something that feels like breathing from outside. The long swells pressing shoreward and retreating, pressing and retreating. The rhythm as old as the moon that drives it, as patient as the stone beneath. The octopus rests in the rock face, and the skin holds its pattern, and the opsins in the tissue wait for a light that has gone, and everything continues in the calm, unhurried way that things continue in the ocean, when there is no one watching and nothing is required of anything. Somewhere in the shallow water, a small bioluminescent creature makes its brief cold light and is gone,